Hello class, and welcome to week 8 of Biology 201. This week we're going to be discussing muscle and muscle tissue as described in chapter 9 of the textbook. As always, please review the objectives as listed in your lesson. So I made this video to help you understand the sliding filament model of contraction theory, anatomy associated with that, as well as the excitation-contraction coupling which occurs during muscle contraction. So let's start macroscopically and look at this muscle attached to a bone via a tendon. Table 9.1 in the textbook describes these different pieces pretty well. So here's the epimysium, which is an overcoat of dense irregular connective tissue, surrounds the whole entire muscle. So this muscle is broken down into a subunit called a fascicle. A fascicle is a portion of the muscle wrapped in a layer of fibrous connective tissue called perimysium, right here. So the fascicle is broken down even further into a muscle fiber or cell, which we will see here in this next picture. So this muscle fiber is covered by endomysium, which is a fine sheath of connective tissue made of areolar and reticular fibers. This muscle fiber is made up of multiple myofibrils as its subunit. And the myofibril is a complex organelle made up of bundles of myofilaments. Within a myofibril is a sarcomere, which is the contractile unit of muscle, which is very important, and you'll really need to know this for this chapter. So a sarcomere consists of different areas here, which we'll discuss. A band is the thick filaments called myosin. The I band consists of thin filaments without any thick filaments overlapping. The H zone consists of thick filaments, and the M line without any thin filaments overlapping here. Brief note on skeletal muscle. The striated appearance of skeletal muscle comes from the repeating series of A band and I bands, which you can kind of see in this picture right here, causing that striated appearance. So let's look a little bit closer at thin filaments and thick filaments and see what they're composed of. Alright, so as previously stated, these thick filaments are made up of myosin, which are a molecule with a long tail, and the important part is the flexible hinge region and the head. So these are essential for contraction. Here's multiple myosin molecules causing a thick filament. So here's the thin filament, and as we said before, consists of primarily actin, but there are a few proteins that are very important to contraction. So the first one is called tropomyosin. Tropomyosin is this rod-shaped protein that spirals along the actin molecules to help stiffen it. And the other protein is called troponin. So it's a complex of proteins which helps to open and close the binding site of myosin. So here is a zoomed out picture of the myosin and actin. There will be multiple, during a muscle contraction, there will be multiple bindings of actin and myosin, which causes the thin filaments to move towards the center, which is essentially what the sliding filament states, which we'll discuss here in a second. A few other pieces of anatomy that we want to discuss before we move on. So the first one is depicted here in blue, called the sarcoplasmic reticulum. So the sarcoplasmic reticulum is smooth endoplasmic reticulum, and it has these interconnecting tubules which surround an entire myofibril, and its primary role is to regulate the levels of calcium, and this calcium is essential for muscle contraction. The next piece is the T tubule. So the T-tubule propagates the action potential into each sarcomere. So T stands for transverse, goes deep into the cell, and the action potential reaches all the sarcomere. Okay. So what is sliding filament model of contraction? So basically it just states that during a muscle contraction, the thin filaments slide past the thick filaments and cause a muscle contraction. So to see this in action, let's look at the series of events that need to occur in order to cause a muscle contraction. So the first thing is we need an action potential. So an action potential comes down from the nerve, down through the sarcolemma, and down into the T-tubule. So the transmission of the action potential into the T-tubule through the endoplasmic reticulum causes the sarcoplasmic reticulum to release calcium into the sarcoplasm, where it's readily available to bind to troponin and actin and cause a muscle contraction. So this calcium binds to troponin as seen here. So calcium goes to the troponin complex, binds, and as it binds, it changes the shape of the troponin, and the troponin tropomyosin complex is shifted off of the actin, and the binding site is then open for myosin to attach to actin. So the myosin head can now attach to actin, and the way it does this is it releases inorganic phosphate and causes a contraction of the myosin head. So after that contraction, the myosin head needs to be released, and the way it does that is by ATP. ATP binds to a myosin head, which causes it to be in the relaxed state and disconnects from the actin molecule. So this contraction process of the myosin and actin is repeated until the calcium level decreases. Calcium is removed from the troponin complex and is shunted back in to the sarcoplasmic reticulum. So once that occurs, the calcium is gone and there can no longer be a contraction, and the tropomyosin blockage is restored, and myosin can no longer attach to actin. So this is essentially what needs to occur in order for a contraction to occur. So now that we can see a contraction, let's come back here and look at what happens to the sarcomere as a contraction occurs. So as described in the sliding filament theory, the thin filaments right here slide along the thick filaments. So as these slide, we need to look at which zones, which bands are decreased in size. So we know that the A band, which is composed of the thick filaments, is not going to change because the thin filaments are the filaments that slide. So the H zone has no thin filaments at rest, but during contraction it will be full of thin filaments here. So the H zone will essentially disappear. The Z disc will also come closer to the other Z disc, and the I band will shorten as well. So we'll quickly review a few of these 6 steps. First thing is an action potential moves down the sarcolemma, down the T-tubule. There's an influx of calcium from the sarcoplasmic reticulum. Calcium is then free and able to bind to troponin, which enables that binding site to be free. The myosin head is bound to actin, contracts. ATP causes the relaxation of the myosin, and this occurs until the signal is gone and the calcium goes back into the cell. So I hope you could follow me through this. If you couldn't, I'd recommend to read the text. I'm also going to put in the helpful hints, helpful links section of the Lesson 8 information posted in the announcements. I'm going to put some animations that you can really look at. So animations are a great way to see this process happen so you could really understand visually how it occurs. So as always, message me if you have any questions, and good luck on the quizzes this week.